I'm Daniel Levine, and this is the Bio Report. One of the underlying limitations of most cancer therapies is the fact that only about 1 to 2% of the drug that's administered ever reaches tumors. That limits the dose because of the potential for unwanted side effects and limits the efficacy by delivering less than ideal amounts of drug to tumors. Shasqi is developing cancer therapies that use click chemistry to activate agents at the site of tumors to reduce systemic toxicity and increase the anti-tumor activity of its medicines. We spoke to José Mejía Oneto, founder and CEO of Shasqi, about the company's platform technology, how it works, and why this can lead to safer and more powerful cancer therapies. Jose, thanks for joining us.
Thank you for having me, Danny.
We're going to talk about Shasqi, its platform technology for developing precision cancer therapies, and how it addresses the dose-limiting toxicity of chemotherapies. Perhaps we can start with the problem of chemotherapies today. It's often said that the hope is to kill the cancer before killing the patient. What's the challenge?
Yeah, the challenge that we saw, Danny, was that, and it applies to chemotherapies, but it applies broadly to most drugs, it's just most prominent with chemotherapies, is that instead of getting the drug to the location that you want, you give drugs that go everywhere. And what you learn in medical school is how much goes to the kidney and the liver and how you should prevent it from hurting the patient. In the specifically to chemotherapies, the issue is that they have extremely damaging toxic side effects. So that's the major issue. It's not that they don't work, it's that they create a lot of havoc when they do their work.
One solution has been to conjugate them with antibodies as targeting mechanisms to deliver a payload precisely and increase the amount of chemotherapeutic agent delivered to tumor cells while minimizing their toxicity. How effective an approach has this been?
It has been an extremely effective approach when you have those antigens expressed in the tumor, meaning when you can separate the tumor from the rest of your body. But there are challenges associated with that approach as well, which has been some of the side effects of these drugs and these antibodies circulating in your body for a long period of time.
One of the challenges, I take it, is finding biomarkers that may not be present in all patients that could benefit from a specific therapy. Shasqi's platform technology offers a way around that. Before we talk about your platform technology and the way it works, though, I thought it might be useful to have you explain two concepts. The first is what's known as click chemistry. What is click chemistry?
Yeah, thank you for asking that. Click chemistry are essentially two chemical compounds that react with each other and ignore the rest of the world. And the way people have described it is thinking as two people in love who meet at a bar and who they exist for each other, but ignore the rest of the sounds and distractions of the bar. So that's a way of thinking about it.
The second concept I think that'll be useful to talk about is prodrugs. I'm familiar with prodrugs, but I take it prodrugs are different. What are prodrugs?
Yeah. Prodrugs are drugs that get activated through metabolic pathways through the body. And protodrugs is similar to prodrugs, except that they're activated by click chemistry only. So they don't get activated by metabolism, different pH or anything, but rather they're activated by the presence of click chemistry. click chemistry reagent in the body.
Does that mean you have to deliver that reagent to where you want the prodrug to be active?
That's correct.
Shasqi has developed what it calls click-activated protodrugs against cancer platform. Walk us through the platform and how it works.
Yeah, just as you described, the objective, the way we see the world, Danny, is we separate targeting agents from payloads. And so the question is, how can we get one of the click chemistry reagents to the tumor? And then can we take a drug, can we take a payload and attenuate it, create an inactive form that only gets activated when that click chemistry reagent is there? And so that is the basis of this CAPAC platform, of the click-activated prodrugs against cancer.
In the case of your drugs, how are they delivered and how are they activated?
Yeah, we have two different generations. And the one that is in the clinic currently as an investigational product works in the following way. We first inject a tumor with a biopolymer, and that biopolymer contains the click chemistry reagent, but doesn't serve any other function that holds it in that location. And then we take a drug, doxorubicin. It's a known chemotherapy that has been attenuated about 80 times, and it's given intravenously through an infusion over multiple days, and gets activated at the tumor.
So you're able to deliver the therapy systemically?
Correct. The therapy gets delivered systemically, but only gets activated at the area that has been injected with a biopolymer.
Given that it's only activated where the injection is, how does this affect the dosing and how much chemotherapeutic agent can actually be delivered to the tumor site?
Yeah, that's a great question. We have shown that you can give more than 12 times the dose. Usually with doxorubicin, you can take 6 doses in your lifetime. And with our approach, we can give about 12 times the dose every cycle, every 21 days. And your second question was, how do we know how much goes to the tumor? We have taken biopsies of the tumor 21 days out, and we have been from the initial dose and have detected active doxorubicin in the tumors. And a second way of measuring how much drug is in the body is by taking samples of the plasma and seeing how much drug is there. And we also detect active drug in the plasma that comes from the amount that was activated at the tumor.
And in terms of the agent that activates the drug, is this something you use universally across the entire platform, or is each activating agent unique to a given therapy?
That's a great question, Danny. Each activating factor can be applied to many different drugs. So you can think of it as kind of a magnet. The magnet doesn't care what it's attached to, but they will react with the magnet of the other sign, regardless. And so that's how click chemistry is working in humans.
You alluded to the fact that this technology isn't unique to chemotherapeutic agents, and they're certainly not the only ones with problems of toxicity. Does the approach have the potential in other indications outside of cancer?
Yes, absolutely. And we have published some manuscripts on that, but Shasqi is exclusively focused on the application of this technology into cancer drugs.
So are you looking to possibly pursue those other indications through partnerships, or?
I think we're looking for pursuing the application of cancer drugs with partners as well. But yeah, I think we're very much interested in expanding the possibilities, and we're showing the way of what is possible with click chemistry. So we hope to inspire others.
Shasqi's lead experimental therapy is SQ3370, which is in development for sarcomas and other solid tumors. What is SQ3370 and how does it work?
Yeah, as I mentioned earlier, SQ3370 is composed of two components. One is a biopolymer that you inject into the tumor, And then the other component is an inactive version of doxorubicin that only gets activated in the presence of that biopolymer of the click chemistry.
I take it has to be injected into each tumor that's present.
That's correct.
Is there an issue with getting access to tumors for treatment?
Yeah. So right now we're focusing on tumors that can be injected. And the interesting part that we found out, Danny, is that when you have a lot of drug concentrated, a lot of doxorubicin specifically, at the tumor, you actually can trigger an immune response. And we have shown in animals that that was possible. And more recently, about 4 months ago, we showed at oncology conference and a couple of different oncology conferences actually that we are able to change the immune cells in the tumors that were injected. And so we hope that those will also help with tumors that were not injected.
So is that to suggest there might be some systemic response once you've dosed an accessible tumor?
Yeah, there may be a systemic immune response that supports the tumor, number one. And number two, as I mentioned earlier, there's also a amount of drug that gets exposed to those other tumors that were not injected.
What's known about SQ3370 from studies that have been done to date?
We know that we have treated north of 30 patients, and so far we know that we have been able to escalate the dose beyond 12 times the molar dose, or the equivalent of regular doxorubicin per cycle. We haven't seen the same side effects that you would find with this chemotherapy. We haven't seen the myelosuppression or the thrombocytopenia that you regularly see, or at the levels that you would see it. And also, it's very, very early days, but we haven't seen cardiac toxicity, even though the total amount of drug that we're giving far surpasses the amounts where cardiac issues would be expected. What's
the development path forward?
Now we have opened a phase 2 study of doxorubicin in soft tissue sarcoma and head and neck patients, and are interested in seeing the responses in these groups, and determining, and particularly in anthracycline naive patients who haven't received doxorubicin before, and then determining the response rates and moving that forward to potentially a registrational study.
Doxorubicin is a well-known chemotherapeutic agent. What's the potential to deliver even more powerful payloads with your platform?
That's a great question. So doxorubicin is a well-known chemotherapeutic agent, and the potential is that you can do the same things that you're doing with doxorubicin with even stronger payloads. And we have actually published some abstracts in conferences showing that that is possible. That we can take a taxane, that we can take an auristatin, things that you could only use with antibody conjugation.
And at this point, are you in development on kind of next generation chemotherapy conjugates?
Yeah, absolutely. We are prioritizing which one are we going to take forward as our next candidate.
And beyond solid tumors, how are you thinking about indications you'll prioritize?
I think solid tumors is actually an area where improvements are needed. That is definitely an area that we're focusing significantly. So I think that's keeping us quite busy at the time.
But I mean, in terms of solid tumors, though, do you have to think more specifically about the indications you'll pursue?
Absolutely. Sorry, I misunderstood the question. So yeah, there's a number of different solid tumors that I think are important to pursue. And there's this new approach that we are developing that is antigen-based. So as you had mentioned with the antibody drug conjugates, they're based on the presence of an antibody, of an antigen in the tumor. And we're very much interested in exploring the capability of our technology to go after those types of tumors that have, like, let's say, a HER2 or some other type of receptor that is unique to them.
Shasqi raised $50 million in a Series B financing. How far will existing funding take you, and how are you thinking about future financing, particularly with the current environment?
Yeah, we were very fortunate to raise that $50 million when we did, and that is providing us a broad runway until 2024. We are currently exploring different options, including partnerships, to further allow us to expand the platform and its potential.
José Mejía Oneto, founder and CEO of Shasqi. Jose, thanks so much for your time today.
Thank you, Danny. Really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.